Welcome to Physiology, Blood. Here is an outline of the topics covered. 1. Whole human blood, IP. 2. Composition and function of blood elements. 3. Coagulation of blood and blood groups. 4. Blood components, manufacturing and storage. And 5. Blood bank operations, requirements, and standards. This presentation was prepared by Mr. Akash S. Jaipai, M.Pharm. Blood. It is a fluid connective tissue that consists of plasma, red blood cells, white blood cells, and platelets. It contains all normal constituents of blood, including RBCs, WBCs, platelets, and plasma. Volume collected is about 450 milliliters plus or minus 45 milliliters. Anticoagulants used include CPDA1, which stands for citrate, phosphate, dextrose, adenine, and ACD, which is acid citrate dextrose. For storage, the temperature is 2 to 6 degrees Celsius. Shelf life is 35 days with CPDA1. Total blood volume is approximately 5 to 6 liters in adults. The pH range is 7. Composition and function of blood elements. Blood is composed of A. Plasma, which makes up 55%, and B. Blood cells, which make up 45%. Focusing on plasma, the composition includes water, 90 to 92%, proteins, 7 to 8%, including albumin, globulin, and fibrinogen. Other solutes are electrolytes, such as sodium, potassium, calcium ions, and chloride ions, along with hormones, glucose, urea, and gases. Functions of plasma. It transports nutrients, waste products, and hormones. It maintains osmotic balance and blood pressure. And finally, it provides clotting factors and B. Blood cells, which make up 45% of blood volume. Component, red blood cells, or RBCs. Structure, biconcave, no nucleus, contain hemoglobin. Function, transport of oxygen, O2, and carbon dioxide, CO2. Component, white blood cells, or WBCs. Structure, nucleated, 5 types, including neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Function, defense and immunity. Component, platelets, also known as thrombocytes. Structure, small cell fragments, non-nucleated. Function, help in blood clotting and repair of damaged vessels. Coagulation, or blood clotting, is the process by which liquid blood is converted into a solid clot to prevent further bleeding from an injured vessel. The steps of coagulation are 1. Vascular constriction, where the blood vessel narrows to reduce blood loss. 2. Platelet plug formation, where platelets adhere and release clotting factors. 3. The coagulation cascade, which involves prothrombin converting to thrombin by prothrombin activator, and fibrinogen converting to fibrin by thrombin. Fibrin forms a mesh that traps cells and forms the final clot. Vitamin K and calcium ions are essential for coagulation. The ABO blood group system. Blood group A has A antigen on RBCs, anti-B antibody in plasma, and can receive blood from A or O. Blood group B has B antigen on RBCs, anti-A antibody in plasma, and can receive blood from B or O. Blood group AB has both A and B antigens on RBCs, no antibodies in plasma, and is the universal recipient, able to receive from A, B, AB, or O. Blood group O has no antigens on RBCs, but has both anti-A and anti-B antibodies in plasma, and is the universal donor, only able. The R factor, or rhesus system, determines whether blood is R positive or R negative. If the R antigen is present, the blood is R positive. If the R antigen is absent, the blood is R negative. The R factor is highly important in blood transfusion compatibility and especially in pregnancy, where incompatibility can lead to a condition called erythroblastosis fetalis. Blood components are separated from whole blood by centrifugation. Packed red blood cells, RBCs, are prepared by centrifuging and removing plasma, stored at 2 to 6 degrees Celsius for 35 to 42 days, and used for anemia or surgery. Platelet concentrate is prepared from plasma by centrifugation, stored at 20 to 24 degrees Celsius with agitation for 5 days, and used for thrombocytopenia. Fresh frozen plasma, FFP, is plasma frozen within 6 hours, stored at minus 30 degrees Celsius for 1 year, and used for coagulation disorders. Cryoprecipitate is prepared from thawed FFP, stored at minus 30 degrees Celsius for 1 year, and used for hemophilia and fibrinogen deficiency. A blood bank is a center for collecting, testing, processing, storing, and distributing blood for transfusion. Operations involve 6 key steps. First, the selection of donor, who must be 18 to 65 years old, have a hemoglobin of at least 12.5 grams per deciliter, and be medically fit. Second, blood collection under aseptic conditions using sterile bags. Third, testing, which includes blood grouping, Rh typing, and screening for infections like HIV, hepatitis B, hepatitis C, syphilis, and malaria. Fourth, separation of components. Fifth, labeling and storage. And finally, sixth, issue of blood and components. Blood bank requirements according to NACO and FDA guidelines include being licensed by the state drug controller, having a qualified medical officer in charge, and possessing proper refrigerators at 2 to 6 degrees Celsius and deep freezers at minus 30 degrees Celsius, along with sterile disposable blood bags and testing kits. Donor and recipient records must be maintained. Regarding standards, the bank must follow the Indian Pharmacopoeia and the Drugs and Cosmetics Act of 1940. Blood must be free from infection and properly labeled. Donor blood collected must not exceed 450 milliliters per donation. Finally, temperature logs and storage standards must be maintained. Here is a summary of all key topics. Whole blood contains all elements, is stored at 2 to 6 degrees Celsius, and is used for transfusion. Blood elements consist of red blood cells, white blood cells, platelets, and plasma, each having a specific function. Coagulation involves thrombin, fibrin, calcium ions, and vitamin K. Blood groups include the ABO and Rh systems, which are important for transfusion safety. Blood components, like red blood cells, platelets, and plasma, are separated from whole blood by centrifuge. Finally, the blood bank is the central facility that collects, tests, stores, and issues